Hello everyone! Today I would like to talk about restriction fragment length polymorphism Rflp. It is basically a technique to differentiate organisms by analyzing the cleavage pattern of DNA. In order to cut the DNA into fragments, specific restriction endonuclease must be present. It is an enzyme that recognizes specific base sequence and cut the DNA into fragments. The more the recognition site, the more number of fragments generated. How does Rflp conducted in the process of DNA typing? Let's say your friends John, Jenny, and Ben were prescribed with clopidogrel. Clopidogrel is a prodrug that needs to be metabolized into active form by CYP2C19. Firstly, extract DNA from the sample taken from them. It can be blood, saliva, or semen. After PCR, add restriction endonuclease SMA1 into the sample solution to cut the DNA into fragments. SMA1 recognizes the sequence CCCGGG and will cut between C and G. If there is no CCCGGG in the gene of CYP2C19, the DNA will remain intact. This means that the gene is CYP2C19*2, which the function is lost. Anyone with this gene will have poor metabolizing function. This is the result. For Jenny, the restriction enzyme SMA1 could not identify CCCGGG site. The DNA remained intact, thus heavier and moved less towards positive electrode. This indicates she is homozygous CYP2C19*2*2, thus poor metabolizer. For Ben, there are two bands, 113 and 110 base pair. However, because of the close proximity of these two bands, you may only see one big band as shown here. This shows that the enzyme is able to cut through its recognition site and suggests that Ben is a wild type for CYP2C19, thus an extensive metabolizer. For John, it exhibits three bands, 233, 113, and 110 base pairs. This is because each individual carries two alleles, one from mother and one from father. This shows that only one allele has the enzyme recognition site. This suggests that John is a heterozygous variant of CYP2C19*1*2, thus an intermediate metabolizer. So this is how Rflp helps in differentiate different organisms or even different genotype in the same species. Thank you.